Hello and welcome to Chapter 5. This is Cell Membrane Structure and Function, and I am again Colin McLeod. The book opens up with talking about a rattlesnake bite and rattlesnake venom. Once it's injected into the bloodstream and maybe surrounding tissues, its action is based in part on its effects on cell membranes, also on proteins which may be embedded in the cell membrane. So as we go through this topic, we'll from time to time come back to what does snake venom do? Why is it so toxic and so damaging? The kid who got bit within hours, his muscle tissue was just completely falling apart. Blood is leaking out of nearby blood vessels to the bite into the surrounding tissue. There's all this bruising and all kinds of terrible stuff. So today we'll learn a little bit more about what happens when somebody gets bit by one of these snakes. Plasma membrane, what is it and what does it do? Number one, main functions: isolates the cell's contents from the environment in a single-celled organism. Everything including and up to the cell membrane, maybe the cell wall if it has one, would be considered the living. Everything outside of it is essentially not living. Also, it's important to maintain certain concentrations of chemicals inside of the cell from the outside. So having some kind of a barrier, i.e. the plasma membrane, between inside and outside is part of just regulating the internal balance of the cell, or homeostasis. 2, regulates exchange of essential substances. So that means it's going to help get the good stuff in and get the bad stuff out. 3, communicates with other cells. In unicellular organisms, that could be for the purposes of exchanging genetic material. In the case of a multicelled organism, that could be responding to a hormone, could be secreting hormones, could be binding to another cell and even causing that cell to do something else or to alter its function in some way. Creates attachments within and between other cells. Well, if you've ever, let's say, tried to pull on your skin, then you'd notice that it hopefully won't come apart because membranes, proteins in the membranes, are helping to prevent it from getting pulled apart. Regulates biochemical reactions. So in some cases, there are enzymes embedded in the membranes, and enzymes can make chemical reactions happen or facilitate chemical reactions. A quick example would be the inside of the small intestine. The inside of it, those cells have enzymes on their cell membrane, and those will help break down any food that you ingest. Structure of a single phospholipid. This is a review. Basics are as follows. They have a hydrophilic head because there is this phosphate group that has, I'm going to focus just on this negative charge here because as a negative charge, it's hydrophilic and is attracted to water. The fatty acid tails of the phospholipid here, they are, well, they're fatty acid. They're just carbon and hydrogen bonds, and those carbon-hydrogen bonds are non-polar covalent bonds. So they are hydrophobic, and they are sort of repelled by water, or water-fearing. Fluid mosaic model. Fluid, what do we mean? It's in constant motion. A fluid is any substance where molecules can flow past one another, so a gas, a liquid. The cell membrane is just like that. It can flow laterally, meaning, let me just circle the membrane here. This is the membrane. Phospholipids in it can move to the left, they can move to the right, forward, backward, and most of the proteins can do the same thing. Mosaic. We say mosaic because it's a mixture of a whole bunch of different things. Mostly we mean proteins. Proteins will float around in a sea of phospholipids, the sea of phospholipids that make up the cell membrane, and they can drift from side to side within the membrane. Some of these are also held in place by the cytoskeleton. More on that later. Last word here, model. We don't know exactly how the cell membrane works, so this is essentially a working model for us to think about any phenomenon that are happening that pertain to the cell membrane. This is just a model for how it happens. Again, science, it's a human endeavor. We don't always know the right answer. In fact, we often don't know the right answer, and we're always improving on what we do know. Sometimes that means saying, oops, we were wrong about something. So this is a model. We don't know everything about it. The phospholipid bilayer. So now we're going to look at just how phospholipids associate with one another to form a bilayer, to form a membrane that can separate the outside from the inside. So those terms hydrophobic and hydrophilic. So here they are, hydrophobic and hydrophilic. These interactions drive phospholipids to form bilayers when in a watery environment. So if you just drop a whole bunch of phospholipids into water, stir them up a little bit, they will naturally associate in a couple of different ways, but this is one of the ways that they will naturally self-aggregate, come together. Why is that? So remember, let's take a look at this diagram right here. All those charged phosphate heads or phosphate containing heads, they're hydrophilic. So they will naturally get surrounded with water molecules. And this is the outside here. So there's water there. And then on the inside in blue down here, there's mostly water there, about 70% or so. We have another layer. That's why we call it a phospholipid bilayer on the inside, and those are all hydrophilic as well. So the two layers, one of them orients outward, the other one orients inward, and then on the inside, so right here where all the fatty acids are, the fatty acids are all hydrophobic, and so they orient away from water. They are not exactly pushed away. We'd have to get into like chemistry and physics. It is, I believe, van der Waals forces that cause them to be attracted to each other. Anyway, you can kind of consider them as, well, fat in oil in water. They are repelled by the water, and so they orient inwards. Okay, so bullet number 1 up here. It's a double row of phospholipids. The proper term is a phospholipid bilayer. Bullet 2, polar or hydrophilic heads face toward the watery environment and the watery cytoplasm. The nonpolar hydrophobic tails orient inwards, and also cholesterol in animal membranes helps to keep them flexible at both low and high temperatures. The phospholipid bilayer is flexible, and that's important because it needs to not break. Cells move around, they bump into things. Sometimes some of them will even kind of like crawl around on the inner surfaces of our body, the inner surfaces of our blood vessels even, and it's important that they be super flexible. Also, it's important because it enables vesicles that are also composed of this exact same membrane in terms of the phospholipid bilayer. It enables vesicles to merge with one another, with other membranes. So when they run into each other, if they've got the right other components, which we won't be getting into, they can fuse with each other, and these phospholipid bilayers just become continuous, right? Here's my really fancy animation. Two vesicles coming near each other, and now they're one big vesicle. Yep, that was pretty fancy. You're getting your money's worth here. Bullet number 2, individual phospholipid molecules are not bound to each other. So again, it behaves kind of like a fluid. Let's take a look at this phospholipid right here. Give you a second to bring your attention here, the one I'm circling here. This one is not chemically bound, as in it doesn't have covalent bonds linking it to adjacent phospholipids on the left or on the right. They're not bound to each other, so they can move laterally. Could go this way by 1 or 2 spaces, could go back to the right. They essentially diffuse left and right. It's like a floating, if you can imagine a 1,000 beach balls floating on a placid lake, and the breeze can kind of blow them around laterally. No way are those beach balls going to get submerged down under the water, but they can move left and right, forward and backward. Let's see. Bullet number 3. Some of the phospholipids have unsaturated fatty acids whose double bond introduces kinks into their tails. So this is referring to the diagram on the right here. And well, firstly, the kinks, these are those kinks that they're referring to. I'm kind of highlighting one right here. And then you can see many other bends in the fatty acid tails. Each of those bending kind of joints is where a double bond is. And the reason for that is associated with angles that are always present when a carbon atom has its 4 bonds, right? Fatty acid tails always have a carbon that is bound to two adjacent carbons and then 2 hydrogens. Anyway, if you introduce a double bond, it causes that sort of linear sort of situation that we see on the left here to become kinked. So where's my arm here? There we go. So to become kinked, a bend forms in it. So you can kind of imagine people standing next to each other, and if all of them start bending their arms out like so or something, they're not going to be able to stand as close together, and so this makes the phospholipid bilayer less dense when there are all these bent fatty acid tails in there. And the cells can manipulate how many of them there are. And I just stated what the last one was. Make sure you take a look at the titles here on the bottom. So on the left, this one has more saturated fatty acids, so it means less fluidity. And that's because the phospholipids pack more densely because all of their or more of their fatty acid tails are just straight. Like people, I'll stand up for you for a second, standing straight with their arms right down next to their body, so they can pack more densely. On the right, more unsaturated fatty acids, so they pack less densely and they can move around more. How is membrane fluidity of the phospholipid bilayer connected to fumbling fingers, being really clumsy when it's cold out? You're always if it's really cold and you're trying to zip up your jacket, you might notice, boy, it's really hard to zip up this jacket when it's really cold. So why is that? At cold temperatures, reading from this first bullet point here, ion channels embedded in the cell membranes of nerve cells are hampered if the phospholipid bilayer stiffens too much. So cells in the hand or in a single-celled organism, if that's what we're talking about, they need to make sure that the phospholipid bilayer does not stiffen too much, in part so that it does not break, but also so that all the proteins in it function normally. We'll take a look over here. At hot temperatures, having more saturated fatty acids helps to keep the membrane flexible and also stable, not too flexible to the point where it just falls apart. So high temperatures, we want the membrane to be more dense so that any movement, because increasing the heat causes all the molecules to move back and forth more. So we want to pack them a little more densely when it's really hot so that they aren't banging around too much. At cold temperatures, right here, having more unsaturated fatty acids helps keep the membrane more flexible. So when you go out, or let me first explain that. So again, they can't pack as densely, as I said. So this helps to make sure they don't pack too densely when it's cold, because molecules are moving less. They're a little more lazy, moving a little slower. So having it be less dense means that they are still going to move enough at those lower temperatures to keep the membrane flexible and keep those ion channels that are part of nerve cells functioning properly. When I say ion channel, right here, that's an ion channel. We're going to come to that later, but you can basically imagine a big kind of thing, a transporter deal, right in the middle of the membrane, and that ion channel would be helping to transport some kind of ion, we'll say Na+, sodium, into or out of the cell. The membrane becomes too stiff, too rigid, then this channel protein here is not going to function correctly. Phospholipid bilayers block most molecules, so we say that they are selectively permeable. So that means that they're not just permeable to everything. They're not just letting everything and their friends in. That would mean that they're not a real barrier if they let everything in. They are selectively permeable. Here are our 4 general rules. 1, small hydrophobic molecules can diffuse through the bilayer. That means they're going to diffuse through it generally much more rapidly. Small hydrophobic. Why? Because most of the membrane here, and you can see, I'll draw it in blue. Yeah, blue is a good color. Most of the membrane here is fatty acids, the fatty acid tails, and they are hydrophobic. Hydrophobic. Hydrophilic molecules generally cannot diffuse through it as well. So example, salts, amino acids, sugars. Water is obviously a hydrophilic molecule. How could water not like itself? Water is polar. It can diffuse through, and the reason that is that it is small. So these are general rules. Smaller molecules, I think that's the next one, right? Next two. There we go. Small molecules generally can diffuse through it more easily, more rapidly, and then large molecules are going to have a harder time diffusing through it. So they're not exactly hard and fast rules. Even if your book says there are, you can just generally think the smaller and more hydrophobic it is, the faster it goes through. The larger and more hydrophilic it is, the more difficulty it will have. This diagram I made myself for you. It took me 20 minutes. I don't know. You're welcome, I guess. I don't know. Maybe you won't like it. So these color codes here. This one, sort of a blue, polar or hydrophilic, and then this one, kind of a yellow. We're going to say that's non-polar, hydrophobic, like butter. It's the same color as butter. So the butter color, hydrophobic. So let's rank all of these molecules in terms of number 1, can pass, and then all the way to 4, can't pass. So generally we'd say the smaller hydrophobic molecules are going to have the easiest time. So here's a little kind of butter colored small hydrophobic molecule, little ones. Those are going to be ranked number 1, can totally pass really fast. Then it would be, I wouldn't expect you to be able to come up with this fine of a distinguishment between can and can't, but next I would personally rank, I would go for these small hydrophilic molecules, so polar, hydrophilic. These blue ones, I would give them a ranking of number 2. So they can pass, but not quite as well as the hydrophobic small molecules. Example, water. Water is a small polar hydrophilic molecule. And if you're out in really dry, cold weather for a long time, or dry, hot weather, your skin will slowly dehydrate because water can get out of it. I'm ignoring sweating for the sake of this example. Lastly, what is going to have the most difficult time is definitely going to be these large hydrophilic molecules. So I'm going to give them a 4. They're getting a 4. And those would be the amino acids, ions, glucose, disaccharides. Those are big, going to have a really hard time getting through. Hey, whoa, look at that. It was like magic. See, the little hydrophobic molecules just diffused through. The little hydrophobic molecules, they were able to make it through. There's one. Yeah, there's one over there too. Now this one didn't quite make it. Oh wow, that was an amazing animation. Okay, so the bigger molecules, what they need is membrane channels. So I've got membrane channel A and then membrane channel B. Let's move them into the cell membrane. They get inserted into the cell membrane by a process we're not going to get into. It is essentially when a vesicle containing these membranes fuses with the cell membrane. Because all the membranes then become one, these protein channels become part of the cell membrane also. Notice I have color-coded the membrane channels as well. This part is polar or hydrophilic, and so that's why it is near other polar hydrophilic molecules on the outside or on the inside of the cell as well. Notice the interior of it is also polar and hydrophilic, which means, let's see if I did this right. Ooh, which means that polar or hydrophilic molecules can pass through it, but nonpolar and hydrophobic molecules would have a harder time because they are repelled. So these channel proteins are somewhat selective, but they are not perfectly selective. It's not like your social security number or anything. It's not that specific. Look at that one. It didn't pass. So the hydrophobic small molecule can't get through there because the interior of this channel protein is hydrophilic, and the hydrophobic molecule is going to bounce off of it. Now this molecule. Notice the interior. I'm going to change colors. Let's try maybe a red. So the interior of this channel protein is all hydrophobic. That serves two purposes. If the interior right here, so this is the inside, I'll just give you an I here for inside of the channel protein. It's hydrophobic, so that is going to mean that it's definitely going to transport a hydrophobic substance. Ooh, look, I was right. Oh, it kind of disappeared there in the middle. Anyway, a big hydrophobic or nonpolar, I'll just write nonpolar molecule got through there. For example, let's say nonpolar, what could that be? Glucose or an amino acid. Right, I just already said that. What else was I going to say? Notice also that the hydrophobic, hydrophilic nature of these channel proteins. Let's take a look at this one first. It is the same as the interior of the phospholipid bilayer. So here, this is all hydrophobic. So these regions all interact with each other, and that helps to keep that channel membrane stable inside of the phospholipid bilayer. Then I'll switch back to blue, and I'm also going to erase some of this because I realize we're getting a little sloppy here. Blue, so this blue square, I'm saying that is polar or hydrophilic. So that means it's going to associate with H2O on the outside, and also it'll associate with these hydrophilic phosphate heads of the phospholipids. Same thing is true on the other channel protein on the left here. So I'll erase this stuff so I can be a little less sloppy. Right here, this is all hydrophilic. So again, that means that this region of the channel protein will associate both with the phosphate heads and with the watery environment and watery interior of the cell. Moving on. I hope you like my animation. Vicious venoms. Some snakes and spider venoms contain the following enzymes. Phospholipase. So phospholipase, that means ACE is a root word that means enzyme. We'll get to that in a later chapter. Phospholipases, they break down phospholipids. So phospholipids make up the membrane, a part of the membrane of every single cell, whether it's a bacteria, a plant, an animal, all animals for sure. So this venom is really broadly toxic. It breaks down the structure, the phospholipid bilayer that separates outside from inside. That'd be like something that breaks down your skin. It's a major, major problem. Next, they also have proteases. Those break down proteins. So all those things that proteins do, we haven't gotten into it yet, may stop functioning correctly if somebody is bit by a rattlesnake. Super dangerous. So that brings us to what do those membrane proteins do? And then we'll come back to, so if you get bit by a rattlesnake, what kinds of functions might it inhibit? So membrane proteins, 4 bullets. Number 1, embedded in the phospholipid bilayer. 2, some proteins can float and drift. Number 3, other proteins are anchored by protein filaments in the cytoplasm. So that would be like the cytoskeleton. So some of them will be anchored to a specific location on the cell, so they will stay there. Bullet 4, many proteins have attached carbohydrates. This is something your book mentioned, so I'll mention it. Those are called glycoproteins. Glyc, that is Latin for sweet. So sweet proteins, I guess they mean that it's got a sugar group attached to it or some kind of a carbohydrate. To point out where those are in this diagram, you can follow my line there. These are carbohydrate side chains. There's one, and then there's a second one here. They occur on some of the proteins. And those are often involved in cell-cell communication, cell-cell recognition. Something goes wrong with one, then it might be part of some type of autoimmune disease. Membrane proteins. Types of membrane proteins. 5 types. Number 1, receptor proteins. 2, recognition proteins. 3, enzymatic proteins. We've talked about them already. They would be on the interior of the intestines, among other places, and they would help to break down food that is ingested. 4, connection or attachment proteins. Those would be in the skin. The skin does not easily break down or tear. If you've ever tried to eat through your skin by, say, grabbing a bowl of food and then just smashing it against your skin on your, on your stomach or your face somewhere, you'll notice it doesn't work. So don't try doing that because you'll not be able to smash food through your skin between the cells because there are all of these connection and attachment proteins that keep it super, super tight. 5, there are transport proteins that carry things into and out of the cells. You do need to know each of these 5 types. Some of them we are going to get into more detail, but not all of them. I'm mostly going to talk here about transport proteins because that's what they like to talk about in this book. Vicious venoms. Some snake venoms contain a toxin that binds to protein receptors on muscle cells. So in this case, it's not necessarily breaking down those protein receptors, but it is binding to them. And this binding, it prevents, here we go down here, it prevents acetylcholine from binding. Acetylcholine, normally, acetylcholine causes muscle contraction, but if it cannot bind to its receptor, no muscle contraction occurs, and this would be local paralysis. So snake venom can cause muscles, some snake venom can cause muscles to basically not respond when the brain sends a signal down to those muscles in the form of acetylcholine that says contract. With this week's lab, you're going to be talking a little bit about molecules moving down the length of a nerve cell that would maybe be going to a muscle. And yes, that's what happens. This impulse or message goes down the length of a neuron. It goes really fast, and when it gets to the end of a neuron, what happens is it'll cause the release of something called a neurotransmitter. In this case, it's acetylcholine, and acetylcholine is released from the neuron cell and then binds to nearby muscle cells. And if it can't bind, then no muscle contraction occurs. Let me make a point here. What I just mentioned about all that is beyond the scope of this test, purely for extra interest. So if you're like, whoa, that was way too fast, you know what? You can restrict yourself to what is written here and in the book. So now we're going to talk about a little bit of chemistry stuff and how molecules move. Firstly, let me remind you, water is an awesome molecule. If you ever want to have a fun conversation with me, let's make it about water. Water is a universal solvent. Very exciting. This means many things will dissolve in it. For example, many of the flavors that are put in soup are the result of things dissolving in water. A lot of the food we eat, it's got water in it, and that's because we can dissolve all these flavorful compounds in it. 3 definitions for you. Number 1. A solute. A solute is any solid that dissolves, for example, sugar, in a solvent, which brings up the question, what's a solvent? Well, water is a solvent. Water is a solvent, for example, a liquid, example, water, that dissolves the solute. So let's look down here. There's a lump of sugar, and then there's the water below it. So the sugar is the solute. The water is the solvent. When you put the sugar in the water, the sugar will dissolve. So that brings us to the last definition here, a solution. A solution is a solute that has dissolved in a solvent. So, following my cursor, here, now we have a sugar solution because the sugar has dissolved in the water. It will then slowly spread out until this state is reached called equilibrium, which we'll talk about a little later. So let me ask you this, a question. In your own words, using the terms we just learned, can you explain why Coca-Cola or another generic soda is a solution? I'll pause the video and try to say it in your own words and try to incorporate as many of these terms that I've introduced now. Pause the video and give it a shot. Okay, if you haven't paused the video, then you missed your chance. Now I will answer the question. Explain why Coca-Cola is a solution. Coca-Cola is a solution because sugar, which is a solute and a solid, has dissolved in water. It has been broken down, disappeared. It has dissolved. It is now not visible in the solvent, which is water. We have now created a sugary solution that we call Coca-Cola. So I said that in my own words, fumbling a little bit with it. And again, I encourage you to try to fumble through some of these new terms because it will help you to remember them. So what do you think? The purple dots are sugar. over time, will there be more or less sugar at point A, and why? So right down here, there's lots of sugar, and this is point A. Over time, will there be more or less sugar at point A? Pause the video and try to answer it yourself in your own words. Did you pause it? Okay, well now I'm going to answer. Basically, diffusion is going to occur, which means molecules are going to spread from regions of high concentration, that is right here, towards regions of low concentration. This movement is random, and so we should say that there will generally be just a net movement to the right towards point A. Some of these molecules will certainly randomly move this way, but more molecules will be moving this way to the right towards point A. Still don't understand? Keep paying attention. Diffusion, that's what I just said. It's a word. Good word, fun word. Diffusion. It is, number one, random. Number two, passive. That means it doesn't require energy. It's just random. It moves. You don't have to spend any energy on it. And it is a net movement. Net movement means overall. Net movement of particles from high concentration to low concentration. If you don't like this word Brownian motion, you can just ignore it. It's a topic of physics, actually. So what we can say is this. Hey, you see that ball there? You could call it a ball, or you could call it a molecule, but it's definitely going to go from high to low. That's diffusion, from high concentration to low concentration. So, again, revisiting the lump of sugar in water example, add lump of sugar to water, sinks to bottom, dissolves in bottom, moving over to the next diagram here where it's dissolved initially. It is going to dissolve, it is going to diffuse away from that region in all other directions. Now, the lab for this week will be super helpful in helping you imagine and via some simulated experiments why we say that this is a net movement. This is a key concept in diffusion and is deceptively complicated, meaning it seems easy, but it's really not that easy. I'll explain why in a second. So diffusion means we're going to move from high concentration to low until this state over here, equilibrium, is reached. And at this state, there's going to be an equal concentration of sugar in every part of the beaker. And if you can't read equilibrium, you'll see it later. So kinetic energy. This is energy for diffusion. Energy for diffusion, it comes from the kinetic energy of random movement of molecules or ions or solutes or anything or solvents, just molecules in general. They're all moving randomly in random directions. So consider, let's just circle one molecule here. Forget about concentration gradients. What direction is this molecule going to move? I have no idea because it's random. It could move away from A or it could move towards A. So let's consider for a moment that let's not imagine up and down and forward and backward. Let's just consider left and right. For the sake of simplicity, it'll be random. So half the time it'll go left, half the time it'll go right. Yeah? So consider now concentration. There's a lot more molecules over here where the concentration is higher. So if half of these molecules, 50%, go right, and then the other half go left, just notice what's going to happen between these 2 points. 50% of these molecules here go left, that's not that many molecules. But if 50% of these molecules are going right, that's a lot more molecules. So that's why the movement, we say it's random, but we also do say that it is very predictably from high concentration to low. Well, that's a little bit hard to read, isn't it? So increasing the temperature will also increase the rate of diffusion. There we go. The rate of diffusion, because the molecules will have more kinetic energy, meaning they'll all just move faster, but it will, of course, still be random. So here's another animation that took me an insanely large amount of time to make, so I hope it's worth it. Again, diffusion is random. This diagram, I like to underline words as I'm talking about them, so you see what I'm talking about. This diagram could be showing solutes like oxygen in the blood or gases like oxygen in the alveoli. So maybe from the air into the lungs, right? So oxygen's got to diffuse across a cell membrane into the lungs, and then into the blood. So it's going to be random. Remember, diffusion is random. Ooh, random. So, oh, there's an arrow, 2 arrows. And notice that about half the molecules on the right have decided to go to the left. So in this case, you could say, what's going on here? This molecule is moving up its concentration gradient. How dare it? It's disobeying the laws of diffusion. Remember, the movement is random, and it is a net movement. The other half go that way. Okay, great. I'm still really confused. Let's look on the other side. Half of those molecules are also going to go left. Half are going to go right. Half are going to go left. Half are going to go right. Okay, so now let's count the number on the left. This is the left. That are going to go across the membrane down concentration gradient. 1, 2, 3, 4. So that's 4 molecules that are going that way, right? Great. On the right side, let's make this a little less messy. On the right side, how many are going to go left and cross that membrane? 1, 2. Because there's a lower concentration of them. There's just less of them there. So we've got 2 going to the left out of the lungs. They're going the wrong way. They're going back into the air. But remember, diffusion is the net movement, circling this word up at the top, net movement of molecules from high to low. So still, there is a net movement of 2 molecules that are going to go into the blood because there's a higher concentration of oxygen in the air compared to the blood. Was that animation worth an hour of my time making it? I don't know. You can email me and tell me I'm crazy. This happens with or without a cell membrane. You see, I just made the cell membrane disappear. Here, I forgot I put that little magical trick in there. Okay, so can you try to basically say what I just said, but in your own words if you want to, or try to incorporate as many of the terms that I have just introduced into what you're saying. What do the following words mean in the context of diffusion? Random, passive, and net. Whoa. Pause the video. It's a hard one. Try to answer it. If you haven't paused it, I'm telling you, I've been a teacher for 11 years. You should pause it and try to say it in your own words, okay? Okay, well, if you haven't paused it, you missed your chance. I will now explain it for you. What do the following words mean? Random. The movement of molecules is random. So from high concentration to low, it's just that in that area of high concentration, randomly, more of them will be going away from that area. Passive, meaning it just doesn't need energy, specifically ATP, but if you just said it doesn't need energy, that's good enough. Net, that means that some molecules are actually going to move up concentration. It's just that more molecules will move down concentration. Okay, I hope you enjoyed that little exercise. So concentration gradient, yay, there's another diagram for it. These molecules on the right, there's more of them there. There will be a net movement towards the left until equilibrium is reached. There's an equal concentration everywhere. Okay, moving on to different types of transport. One broad category of transport is called passive transport, and it essentially refers to diffusion, but there's a couple other types of diffusion. So it's diffusion, meaning it doesn't require energy. That's why we call it passive. It requires no energy. The molecules in the cells can be sitting on the couch. It's still going to happen because it doesn't require energy. It happens in any situation. Could be inside cells, could be outside of cells, could be across cell membranes, in the ocean, in the atmosphere, in space, or even on another planet or in an alien's body. That's going a little too far. Bullet number 2. When diffusion occurs across a cell membrane, it happens without the cell using energy. So we call it passive, no energy used. Great. Passive transport, no energy used. This is the definition of passive transport, by the way. It is 2 things. No energy used, and it is down concentration gradient. So yeah, right, by diffusion, because diffusion is always down concentration gradient. This is just another diagram down here on the left showing you that if there's more molecules on the left, passive transport or diffusion across this membrane will result eventually in a net movement to the right and eventually in this state called equilibrium. I'm not going to write it out for you again. Passive transport, diffusion. First kind, and now this is a subcategory of passive transport. It is a subcategory of diffusion, sort of, basically. You won't be quizzed on the difference. Simple diffusion. It's this simple. So number one, bullet one, lipid-soluble molecules, basically meaning, generally meaning non-polar molecules, hydrophobic molecules, lipid-soluble. Lipids are fats. Example, vitamin A, E, oxygen, carbon dioxide, and also very small molecules like water. They can diffuse directly across the phospholipid bilayer. They don't need to pay a toll, nothing. They will move from an area where there's a high concentration to an area where there is a low concentration. They'll simply diffuse across the membrane. This is called simple diffusion. Facilitated diffusion is when there's a little bit of a helper protein that is going to be involved in doing this. So facilitated diffusion is a subcategory of passive transport. Bullet one, water-soluble molecules like ions, amino acids, and sugars, they cannot pass easily through the phospholipid bilayer on their own, even with a concentration gradient. So let's look at the bottom left diagram, and I'll circle all those glucose molecules. Look, there's so many outside, and on the inside, this is the inside here, right? There's so few. So they should just diffuse in. Well, they can't because they are too hydrophilic and they're too large. So they can't just diffuse through the cell membrane. They require a helper to facilitate their transport. These are membrane proteins that allow these specific solutes to move through their hydrophobic region of the membrane. And my very time-costly animation of hydrophobic interiors of these transport proteins, you can look back at that. The insides, let me circle this right here, the insides of these would likely be hydrophilic, so that glucose, also hydrophilic, will not be repelled. Next. So there are three types of protein channels that are involved in facilitated diffusion in this course. You take a more advanced course and it'll tell you about a whole bunch of other kinds. It'll be really fun. I'm not being sarcastic, even though I sound like I am. It will be fun. Carrier proteins. Number one, bottom left of this diagram, glucose is being transported by carrier proteins. Number one, what happens with a carrier protein? It carries the thing it's transporting. So here, glucose, it's going to carry it. So if it's going to carry it, that means it's got to touch it. Meaning, in biology, we say bind to it. So it will bind to the molecule being transported. Good word to remember, bind. Number two, it will change shape in response to binding. So it will, without any input of energy, revert from position one, this is position one, to position two. And that's because all atoms and molecules are always kind of jiggling like so. And so the energy supplied by just kind of the ambient temperature, which is the kind of energy, causes these proteins to just kind of like go jiggle back and forth. So it's like, got a glucose molecule, moving it over here. Oh, I don't have a glucose molecule. Oh, now I got another glucose molecule, moving it over here. That might have been a weird explanation. Let me add one more thing. This will go in either direction. It is simply based on concentration gradient. So if I erase all this stuff again, and again say, see, high concentration here, that is going to determine that the direction of transport will be into the cell. But if for some reason this cell had a lot of glucose inside of it, then it would diffuse through these same proteins in the opposite direction. Concentration is what determines the direction of movement. Why? Because it's still a kind of diffusion. All right. Number two, channel proteins. These are, number one, specific. And then by specific, I mean pretty specific, but they can be fooled. Number two, some remain open while others must be stimulated to open. So this is Another example of kind of the nervous system. The nervous system, if you've ever seen any kind of TV science about like there's electricity or something involved in the nervous system, well, there kind of is. And a bunch of charged particles on one side of the membrane will be all of a sudden they're let in through one of these channel proteins when it is stimulated to open, and that is why we can respond really fast, because when one of these channel proteins open, the movement of these charged particles down their concentration gradient, it's super fast. That's why we can do things like this. Watch. Open, close, open, close, open, closed. Pretty fast. That's because of channel proteins. Last one. Aquaporins. They are the same thing as channel proteins, except that they're for water, and they are, in all examples I have ever seen, they are always open. So if they're in there, water is going to be going through them, and it's going to be going down its concentration gradient. So from wherever there's more water to wherever there's less water. Passive transport, osmosis. In plain words, osmosis is the diffusion of water across a selectively permeable membrane to reach equilibrium. So this is a kind of a tough one, but it sort of seems simple, and that's usually why it's tough. It's because students don't focus enough on it. So it's not that simple. It is diffusion, but it is we only say osmosis if the thing that is diffusing is water. And we also only say that it is osmosis if this thing called water is diffusing across a permeable membrane. Only if it is diffusing across a membrane. So there's got to be a membrane there. It occurs without energy. That's because it's diffusion. You can think about the directionality of water movement in two ways. Let's all take a deep breath because this is kind of annoyingly confusing. Yeah. Number one, water moves from an area of high water concentration to an area of low water concentration. That's diffusion, isn't it? But often in biological contexts, we will say it in another way. Number two, water moves towards an area of higher solute concentration. So you can think water follows salt, and the terrible example I give, and don't go out and do this because it's very cruel, but we're all kids once, and I don't know, kids can be cruel. Slugs, right? What happens if you pour salt on a slug? It's not pretty. The water inside of the slug just diffuses out of the slug to where all that salt is, because the salt is pure solute, so that the water inside the slug's body is going to diffuse out of the body, resulting in, I would imagine, a terrible death for the slug. So they will both give you the same direction of water movement. So basically, whichever way of thinking is easier for you, you can generally just do that. Osmosis. So what is happening here on the left, we start out at this kind of high level. What is it, about here or something, like where it starts off? No, it starts off about here. And when in the beginning of this animation, it's pure water. So that's the highest concentration of water that you can get, right? It even says that at the bottom here. High water concentration, pure water. And they're saying that this membrane here is selectively permeable. Now, if we look what's going on, it looks like it is only permeable to water. So only water can diffuse. Next, how is it that on the right-hand side, this is a lower concentration of water? It's weird to think of water as a concentration, because usually we're doing things like adding salt to water, and then there's still the same amount of water, and now it's a 5% salt solution. So think about it this way. If this on the right-hand side here was once pure water, as soon as you add sugar, the green circles, as soon as you add sugar, it's not 100% water anymore. And so what that means is some of these water molecules are going to get kind of involved. You could say, for the sake of explaining, that they fall in love with the sugar molecules and are simply unwilling to move to the left. So all these water molecules on the left, there's more of them, and they move to the right, diffusing down their concentration gradient. The scientific term for why it is that adding solutes to a pure water solution is involved in osmosis, and it is this. water molecules associate themselves. They actually will bind to some solutes, forming hydrogen bonds. So when they bind to those solutes, they stick with them. They stick to them. They literally stick to them. So they can't go across to the left across this membrane because the sugar molecule itself cannot go across. So it can't go across. The water molecules on the right, less of them can go across because they associate with the sugar molecules. Okay, semi-permeable membrane, same example. Moving on. Tonicity is a term you need to be familiar, and it refers It refers to the concentration of solute relative to water concentration. So we're talking about solute concentration now. Usually in reference to how conditions inside and outside of a cell compare to each other. So usually we use this term only when we want to talk about how different is it outside the cell compared to the inside of the cell. If the solution a cell is floating in or sitting in, whatever, if it has the same concentration to the cytoplasm of the cell, the solution is said to be isotonic. Iso, this root word means the same. It's said to be isotonic. If the solution has more concentration than the cytoplasm, the solution is said to be hypertonic. Hyper means kind of like above or higher. It's a good one to know if you're going to take 1,000 biology classes because it keeps coming back. So hypertonic means above, higher concentration compared to the inside of the cell. If the solution has less concentration than the cytoplasm, the solution is said to be hypotonic. Hypo, root word, means below. So looking at what happens to both animal and plant cells in these three environments, every single book that I have ever had, and that's probably about 20 that I've looked at, used to teach, always uses this diagram. I don't know why, kind of weird. But here it is. On the top, let's talk about animal cells. First, in an isotonic solution, this red blood cell, which I will now abbreviate to RBC, is perfectly happy. The same amount of water is going in as is going out. The plant cell, on the other hand, is flaccid. This would be similar to you not watering your house plant. I don't mean to accuse you of anything, of me not watering my house plant, and then it becomes droopy because it is flaccid. So why is it flaccid? It's because the central vacuole here is not filled with water, not pressing up against the sides of the cell membrane, which then presses against the cell wall. More details on this a little later. So if that was a little confusing, I'm going to get back to it in a bit. Hypotonic solution. The way that I still to this day, is that true? It's a bit of an exaggeration. I just have it memorized now, but hypotonic, I think of a hippopotamus, because they're big, round, and fat. They're not fat. They're just big and round. That's not fair to hippopotamuses. Hypotonic. What happens to animal cells is they blow up like a hippo. Hypotonic. Because more water is diffusing in by osmosis across the semi-permeable cell membrane, and the cell, if it's an animal cell, will expand as much as it can, and then it will lyse or burst. This is their animation or their cartoon for the cell bursting. In the plant world down here, this plant is absolutely just happy as can be. So I almost drew a smiley face, see? Now it is a smiley face. That's terrible. I'm not going to waste your time. Okay, why is this plant happy? Because it has now been watered. If it's a house plant, it's now going to be right up there like this, because more water is flowing in at any given moment than is flowing out. This means that water flows in, fills the central vacuole, which then presses against the cell membrane, which then presses against the cell wall, making it full like a balloon, or like this, like that. And that's the environment the plant cells are happy in. We say that the cell is turgid. You do need to know that word. More on that later, though, because this is really important for plant cells. Hypertonic solution. Hypertonic solution, let's go back to plant cells for a moment right here. This is fatal to a cell. Something called plasmolysis occurs. Too much water diffuses out of the plant cell because the outside external environment has a higher concentration of solutes. So water diffuses down its concentration gradient to where there's more solutes. And what happens is the cell membrane and central vacuole, they shrink, and they shrink so much that the cell membrane tears away from its cell wall, and that tearing is irreparable. There's all these proteins in that plant cell membrane that were attached in very specific ways to the cell wall, and once they've been torn away, it cannot be repaired. So this is lethal to a plant cell. That's why you need to water your plants. I mean, not accusing you. That's why I need to water my plants. And now a more detailed explanation on what this term turgor pressure means. Turgor pressure in plant cells. So on the top, let me just reference these pictures here. On the top, this is a plant that's happy and it's got a lot of water flowing into it. And then the picture on the right is what a plant would look like if all of its cells are getting enough water. It's springy and it's just saying, ha, 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 hello, I'm turgid. And then the picture on the bottom left, this is what happens when a plant cell is not getting enough water. Let me reference the cell membrane here, or highlight it rather. Notice it is pulled away from the cell wall. This is the cell wall. So the cell membrane and cell wall pulled away from each other. This is way bad. This plant is probably going to die, or if it's not dead already. And notice down here, the leaves are wilted, or they're limp or flaccid. So now let's go through the details of it, largely in reference to the central vacuole, aquaporins, and the cell wall. So the central vacuole has lots of aquaporins. What that means is, I already wrote it down. The central vacuole is a high concentration of dissolved substances, solutes. So water is attracted, saying it in human terms, anthropomorphizing a little here. Water is attracted to that central vacuole environment because there's lots of solutes there, and that means there's a low concentration of water. So water diffuses in first from the environment into the cytosol, and then from the cytosol into the central vacuole. The central vacuole is hypertonic to the cytosol in the environment. Hyper meaning more solutes. So water diffuses from here, where there's lots of water, because I've been watering my plants, very, very responsible. So there's lots of water in the environment, and it's going to diffuse into the cytosol, across the cell wall, across the cell membrane, and then, as I said, there's lots of aquaporins in the central vacuole, so it easily diffuses into the central vacuole, filling it up. The central vacuole pushes on anything around it, and eventually starts pushing on the cell membrane and then cell wall, and it's just, and that puts pressure on the cell wall. That's called turgor pressure. And I've provided a link on Blackboard, providing they have not stopped using Flash, which they keep threatening to do every day. Should be still accessible though. And it shows water moving into and out of cells in cartoon format. Take a look. 3. I already said that, turgor pressure. Moving on. Active transport. This is when the cell has already got a lot of the good stuff, whatever it is. Maybe it's sugar. Okay, we all like sugar, right? Don't we? Oh. The cell has a lot of glucose in it already. So the cell should be happy, but it's a greedy cell. It has a high concentration of sugar, but it wants even more from the environment that has very low concentrations of sugar. So diffusion will not work, because diffusion would mean that that high concentration of sugar inside the cell, sugar will start diffusing out of the cell. What we want is to move from low to high concentration. So the cell's got to expend some energy. So here's your formal definition of active transport. It is the movement of molecules up their concentration gradient across a membrane, and it requires energy. So it's not the same as diffusion because it's going up concentration. It is not passive transport because it uses energy. That's why we call it active. It uses energy, particularly in the form of ATP. And if you're thinking, what's that? That's such an annoying word. It's just like 3 letters. Well, for this course, let's just satisfy ourselves with thinking of it as the money of the cell. If you've got it, you can make things happen, all right? And you can email if you want more details, but you'll probably wish you hadn't. Sometimes a cell needs a certain substance, even though it has a lot already, so it's got to spend energy. The cell expends cellular energy to move that substance against its concentration gradient, so up concentration gradient from low to high. Example, absorbing rare ions or sugar from the intestines. Your intestinal cells will do this. Plants use it to pump minerals into their roots from the soil. This is one way plants compete with each other. They can't like just go somewhere else if they don't like it. In this area, they can't move, get on a plane, go somewhere, can't do it. So they're stuck in their rooty, dirty environment. So the way that they've got to compete with their neighbors is they've got to try to pump into themselves as much of those rare ions that they need as possible. They pump as many into themselves, whatever. They pump as much into themselves as possible, and they're willing to spend that cellular money on it because it's just so rare. Now, back away from the strange imaginary world of ATP as money, we're going to look at a biological example. So, there it is. Calcium pump example. So this pump binds to calcium. Binds. That word is important to use. You can't just say it takes it. Let's use the more technical words you use when answering, say, a test question that requires you to write. The more technical words you use, the better. So it binds to the calcium. The transport pump binds to the calcium. Let's now go down to the picture here. Calcium. So here it is, bottom left. Calcium binding site. So the calcium is going, like my sound effects? I don't know. I've been working on them pretty good. So calcium binds to the transport protein. Then ATP, the dollar of the cell, right? It's going to say, hey, do you think you can get my little buddy calcium into the cell here? And then the protein pump says, well, if you bind to me, then yes. ATP binds as well. And this input of energy from ATP is going to make the blue transport protein pump here change shape. And it's going to have to do some work like, and it'll release the calcium on the other side of the cell. Finally, you can see right here the ATP is still there. Finally, it will be released in the form of ADP and phosphorus, and basically, let's not get into it because your book doesn't explain it all. Email me if you want to know about this ADP and P. Yeah, you'll wish you hadn't again. Okay, yeah, okay. Check, did everything. Another form of active transport is endocytosis. If you get as excited about Latin root words as I do, then you'll love this. Endo means inner, cyte means cell, osis means process. So the process of taking things into the cell. Specifically, a couple of kinds. Number one, some substances a cell needs, they could just be too big to move through the cell membrane or even through a transport protein, so can't work, won't work that way. Well, two, some cells use a process called endocytosis to engulf larger substances. There are 3 types of endocytosis. Pinocytosis, receptor-mediated endocytosis, and then the last one. Don't need to write them down now because we'll get back to all of them, but you do need to know that these 3 types, these 3 terms here are types of endocytosis. So active transport, endocytosis, a kind of endocytosis is pinocytosis. This is basically cell drinking. So how does it work? Pinocytosis is when a cell brings in fluid from the environment. So first, this is the cell membrane here in yellow on top. So you see what I'm looking at here? That's the cell membrane. First, a dimple forms. Some books will call it an invagination, like so. And water will then fill that space, or whatever fluid's out there will fill that space. It then enlarges, so there's a deepening pit, and then it is also going to pinch inward here, Finally, it pinches off at 3. It pinches off here, forming a vesicle that has fluid in it. Then that is taken in. The cell now has more liquid inside of it. Huzzah! I'm pretty happy. Active transport receptor-mediated endocytosis. So receptor is going to refer to a protein receptor. Mediated means the protein receptor is going to sort of facilitate the process of taking in a big something or other. So RME, receptor-mediated endocytosis, right here, occurs when the cell takes in specific substances that are too large to be transported through the cell membrane or through channel proteins. So let's look at the picture here, right here. So we're saying that these green circles, they're too big to get in any way other than some kind of endocytosis. We say that it is specific because, here we go, the cell has to recognize or bind specifically to that substance before endocytosis can begin. So let's look right here. If you follow the arrow, the blue structures there that are kind of Y-shaped, those are the receptors, and they are very specific. They will basically only bind to one type of molecule that they will take into the cell. So then, how does it happen? Number one, this occurs in coated pits, coated with receptors that are specific to specific substances. So these coated pits, they've got receptors for specific substances. 2, the receptors bind to that substance, and then the pit deepens, eventually pinching off to form a vesicle in the cytosol. So 2 is referring to this. The pit deepens, and then 3, it will eventually pinch off, forming a vesicle that will now be inside the cell. Hooray, we're done. 3, though, just a couple of examples. Sometimes taking in hormones that are just too big to get in any other way. Also taking in those, we talked a little bit about LDL, HDL, the transport of cholesterol. It's a big molecule, so cholesterol containing lipoproteins will get into cells like this as well. Phagocytosis. Phago. This refers to the term eat. If you're interested in the Latin, it might help to remember. Cyte means cell, osis means a process. So the process of cell eating. Oh, it's written right here, right? See? Cell eating. Active transport requires energy. Here's your definition. Phagocytosis is used when a cell, an amoeba, white blood cell, takes in a larger substance, or not a larger substance, but like a big one. Sometimes even a bacteria or a small protist. Protist is a eukaryotic single-celled organism. So this is cell eating. One, bottom right. This is a good picture. So the large fella here is eating the orange small fella, and it is essentially just going to wrap itself around the smaller one and then pinch off, and then the smaller one is inside, and then it's just like, and it'll break it down. How would it break it down? Review lysosomes. Lysosomes would then get fused with that poor little cell that's been phagocytized, and lysosomes would then release all these horrible substances that'll break it down, chop it up into whatever favorite dish this cell likes to eat. Probably carbohydrates and proteins. So now the details of it. A, this one here, phagocytosis, some type of a food particle. It could just be food, could be a bacteria. Firstly, the cell is going to essentially wrap its membrane around that smaller cell or smaller food particle. Eventually, the cell will pinch off its cell membrane around the food particle. Now, the food particle is inside of the cell, and if it is going to be digested, then it would fuse with a lysosome. Remember, lysosome. And if this lysosome fuses with it, it's going to break down the food in there so the cell can eat it. There, I'll try to put as long as it's still on YouTube, a 45-second video of an amoeba engulfing a paramecium. It is quite cool. Exocytosis. So this, you can maybe guess, is the opposite. So exo means out, cyte means cell, osis means process. So the process of exiting the cell by this way. So bullet one, exocytosis. It's a way for cells to release large substances quickly. Remember, it's active transport, so it's going to require a bunch of energy. What are we doing with it? Could be used for getting rid of waste products. Not as common a thing, but more often to secrete cellular products like hormones or antibodies. And a white blood cell that is secreting antibodies can secrete by this method like 1,000,000 every second. So this is a really fast way to release a whole bunch of stuff. How does it work? Number one, we have a vesicle that has pinched off from, say, the Golgi apparatus. Maybe it pinched off of the rough ER or the smooth ER. Anyway, it's got something in it that the cell wants to release into the environment. So this something right in here, we'll call A that something. It wants to release substance A. The vesicle containing substance A is moved to the cell membrane. How is it moved there? Well, if you're curious, it's moved on the cytoskeleton, and that requires a huge amount of energy, but it's moved there somehow. And then it fuses with the plasma membrane. And once that fusing occurs, this is where that fusing has occurred, the contents of the vesicle are going to be released into the environment. Substance A has been released. Oh boy. And this right here, this A here is a real electron transmission micrograph of something being released. Pretty neat. Electron micrographs give us a huge amount of detail of cellular processes. Notice also on this electron micrograph circling the plasma membrane label here, that the plasma membrane is super fluid. It can bendy all around like crazy without breaking. So that is another feature of the plasma membrane that is very useful. Exchange of materials across membrane influences cell size and cell shape. Cell seashells by the seashore? Yeah, okay, I can do it. Bullet number one, hypothetical cell. So 8.5 inches in diameter. That's a monster cell. It would take 20 days for oxygen molecules to diffuse to the center of the cell. That's just too long. I mean, have you ever tried holding your breath for 20 days? It's impossible. I tried. I lasted about 1 minute. So that would be way too slow. The cell will suffocate. Next, the same amount of time is going to be required to get carbon dioxide out of the cell by diffusion. So that's why big organisms have all these tissues and organs to facilitate the removal of toxins and the intake of the good stuff, like oxygen. But even single cells, they can't get huge, and it's for very specific reasons. So as the diameter of a circle cell increases, its volume is going to increase more rapidly than its surface area. So, pardon me. Let's look at volume here. So when we go from this, let's look at radius also. So if the radius is one inch or something like that, then the volume is going to be 1x as well. Okay, fine. And then let's just say we increase it by twofold. Oh, yeah, right. Also, got to mention this too. The surface area is also 1x. So 1x radius equals 1x volume equals 1x surface area. Next, if we increase the radius by 2, so we double the radius, then if you do the math here, if you calculate the volume of a sphere that has had its radius doubled, then you're going to find that the volume has increased by 8-fold. So that's a big problem, because that's a lot more volume, a lot more stuff inside there that needs oxygen to get to it, and may also be producing carbon dioxide or other waste products that need to get out. So you have this huge oxygen requiring volume and waste producing volume, but notice the surface area has only gone up by 4x. So the next increase in radius here leads to 27 times greater volume and only 9 times greater surface area. So there's just not as many there's not as much surface area available in this hypothetical giant cell, not as much surface area available to service it with things that it needs and can only get by diffusion. So why cells are small? Again, referring back to the elephant. Yes, one reason the elephant consists of many cells is that it needs a whole bunch of different cell types to form different tissues and different organs. But the rate of diffusion is just so slow, it makes having a giant cell impossible. So cell 1 and cell 2, they have the same volume. You can see, right? Look, they're the same volume. I mean, like 1, 2, 3. If we look on at number 2, right? 1, 2, 3, 4, 5 of these little squares up. Okay, and then this one's like 5 of those little squares up as well. So they're all the same dimension. So they must be the same volume as well. But if we have this kind of model, as in model 2, look at all of those internal surfaces of those little cubes. Each of those serves as a potential site where oxygen can diffuse in and carbon dioxide or other waste products can diffuse out. So it's no problem for version 2 here for these little cells to get rid of all their waste and get in all the oxygen, because everything has to just diffuse not that far at all. Big old cell like this, right in the middle, if this is a CO2 molecule in there, diffusion is just way too slow. So vicious venoms, coming back to that, they contain phospholipases, which can break down cell membranes. Phospholipases, so remember phospholipids, they can account for about 50% of the cell membrane. So if you've got an enzyme, that's a kind of protein that breaks stuff down. So this kind of enzyme, a phospholipase, will break down phospholipids, and that's not good because, for example, if this starts happening to red blood cells, they transport oxygen. You're gonna become anemic, not enough oxygen being transported, and if that snake bite got enough of that venom into your blood, can cause anemia rapidly. Bullet 3, muscle cells, that'll simply break down the muscle cells, so, because we're breaking down the phospholipids that form the cell membrane, and so it leads to necrosis. You don't need to memorize that word, but it's, I think, kind of a cool word anyway. So death of tissues, death of muscle cells, and here's that picture showing that. It's not pretty. Last one, blood vessels. That will, if blood vessels, meaning the veins and arteries of the body, if their walls are broken down, that can lead to internal bleeding. So some of these venoms and snakes, spiders, it is really nasty to deal with. Just about to the end here. Antibiotic resistance and cell transport. So firstly, antibiotic resistance. Antibiotics are any compound that will kill bacteria. They kill bacteria in a variety of ways. I'll explain that in a moment. Let me first give you an example of osmosis. So number 1, let's talk about the cell wall and osmosis. Remember, bacteria generally have a cell wall. I'm going to circle it so you know what I'm talking about, right? There's a cell wall. It's the outermost layer here. It's the outermost. It is strong. I'm just going to make it really clear. That's a cell wall there, yeah? Now, the inside of a bacterial cell, and pretty much all cells, is going to be hypertonic to the outside environment. So hypertonic means lots of solutes inside the cell. That's going to lead to H2O diffusing into the bacterial cell. So the cell membrane, so this is the cell membrane. Locate what I'm circling here. This is the cell membrane. There's going to be pressure on the cell membrane from all this H2O that's flowed in, putting pressure on that cell membrane. The cell membrane expands, and then it's putting pressure on the cell wall, and the cell does nothing. It's totally happy, totally fine, because the cell wall helps to maintain the structure of the bacteria. The bacteria is completely fine. So what an antibiotic does, and this is just one kind of antibiotic, there's many other kinds, but one kind of antibiotic, what it does is it will weaken the cell wall. So it's going to weaken it. Let's draw some weakened cell walls here. Weakened. weaken cell wall. I hope you appreciate the sound effects. Weaken cell wall. Weaken cell wall. Weaken cell wall. Get it? The cell wall is weak. So now what happens when this bacterial cell is just kind of hanging out? Its cell wall turns out it's just weaker than normal. So let's highlight what we already just talked about. H2O is pretty much always diffusing at some rate into bacterial cells because the inside of a bacterial is hypertonic, more solutes inside the bacterium. Now, if there's all this water inside the bacterium, it's going to what? It's going to put pressure on the cell membrane. Then the cell membrane is going to put pressure on the cell wall, and normally bacteria would be totally happy, but oh no, its cell membrane is too weak. Its cell wall has been weakened, so it literally explodes. The bacteria will lyse. It will explode, and therefore antibiotics will rapidly clear up a bacterial infection because those bacteria inside whatever infection it is are going to die like crazy. But bacteria are evolving resistance rapidly. Review from what we've already talked about. I'm gonna first erase some of this stuff so we can see what we're talking about again. Go a little crazy with that pen sometimes. Sorry. Here we go. So this thing right here, this is a plasmid. Plasmids often carry antibiotic resistance genes. Why do they carry them more often than the bacterial chromosome? Remember, bacteria have chromosomes. Here's the bacteria chromosome. Why is it often that the plasmids, that's another kind of DNA, more often carry antibiotic resistance genes? Because they can be traded and exchanged between bacteria. And so if one bacterium happens to have a mutation in a plasmid that enables it to survive the attack of the antibiotics, then it is going to be more likely to survive, reproduce, have a bazillion trillion offspring, literally like a bazillion trillion. And all of those offspring will have that plasmid, and they can transfer that plasmid to other bacteria, even different species of bacteria. Kind of scary. So for mutations happen more often in the plasmid. Here's what one of those mutations will do. It will cause this bacterium to be able to produce a membrane protein pump, cell transport. It's going to be number 3 here, active transport. So DNA on the plasmid results, if we have the right mutation, results in, let's look here, the insertion of what's called an efflux pump. And that's basically just what we've been talking about with active transport, an active transport pump that pumps the antibiotic itself out of the bacterium. So this yellow molecule here, that's what they're using to say this is the antibiotic. So you've gone to the doctor, the doctor's given you some antibiotics, you take them and they get introduced to where the bacteria have infected you. They diffuse into the bacterium, but then the bacteria just pumps them out. And eventually they get broken down randomly outside the cell because eventually everything gets broken down. So these bacteria are totally immune to this antibiotic. This is a summary of transport mechanisms. This is more used for review, so I will not be referencing it, but I'd encourage you to take a look at it. It'll help you to organize your thoughts. All right, that is all for this lecture. Sorry it went a little long, but the other one went a little short, so we try to spread it out across a couple of weeks too. So anyway, hope you enjoyed the lecture. Have a nice day.